So thank you for having me here. I'm Shelley Bennett. I'm a PhD student at the University of California, Santa Barbara. I'm in the Riparian Invasion Research Lab, and today I'll be talking about ecology and control of shell boars. So before I start, I just want to thank some people, my advisors, people in my lab who helped me with field work and collecting trees in the field, all of my undergraduates who helped me process all of my samples and made this all possible, and then of course my funding, and thanks to the LA Urban Center. Okay, so Leon did a great introduction for me for the shell boar, so I'll try and go over this pretty quickly, but these two invasive species are now present in California, the Polyphagus and Croceo shell boars. They're actually part of a 5-species complex of beetles that are all morphologically identical but genetically distinct, and we only have 2 here now in California. So they are ambrosia beetles. They're similar to bark beetles. They live inside of their host trees, but what sets them apart from bark beetles is that instead of feeding on the tree itself, they actually feed on symbiotic fungus that they inoculate into the tree. And so here you can see this is what we call a gallery. The beetles bore into the tree and they inoculate their fungus, and it feeds the larvae and the developing beetle, and the adult beetles. So they are both invasive in California. They're spreading rapidly geographically, and they also have a very wide host range. And they are vectors of Fusarium dieback, which is the disease caused by their symbiotic fungus. And I just want to give a little background on their biology. So the way they utilize host trees is that a mated female will find a host tree. She'll bore into the bark and start creating her gallery, that system of tunnels. She's going to inoculate her fungus right away. They carry up to 3 different species of fungus, but usually it's the Fusarium and Graphium species. Those have been shown to provide the food source for the beetles. Then after about 5 days, the female will lay eggs. Those go through 3 larval stages and pupate inside the needle gallery, and then the brothers and sisters actually mate with each other. The males never leave the gallery, and then the females will leave to find their new host. And so it's not really the beetles themselves that are causing most of the damage, it's actually that Fusarium species, the fungus. It's causing this disease, Fusarium dieback. And so the way this works is that the Fusarium is inoculated into the gallery, which is usually beneath the cambium layer, right in that heartwood of the tree. But you can see right next to the heartwood is the xylem. That's the tube that allows water transport from the roots to the crown of the tree. And so basically the fungus physically blocks water transport through the tree. Therefore the leaves are no longer getting water, it can't photosynthesize. So that can lead to branch dieback. You can see in this box elder, all these top leaves are dying back. Or it can lead to entire host mortality. This is a bunch of willow trees that have died in the Tijuana River Valley. And like I said, they have really wide host range. So they've been detected in over 200 species of trees, but not all of those are suitable hosts for the beetle. But if the beetle does successfully inoculate its fungus, a tree can become susceptible to the fungal disease. 64 of those are reproductive hosts of the beetle. As Leon said, it's a tree where the beetle can successfully reproduce within. And those include our important agricultural trees like avocado. You can see here is this characteristic sugary excretion, and then this also includes important urban trees like oaks and sycamores, and also what my lab is interested in is the riparian species like willows, cottonwoods, maples, and alders. You can see here is a red willow. Hopefully you can see this is some of that staining, which is characteristic of the willow's symptoms. And like I said, my lab is really interested in this riparian aspect because in the Tijuana River Valley there was actually a huge dieback event. This was a photo taken in May 2015, and they had just then detected the Croceo shell boar there. And just less than a year later, in February 2016, the same area looked like that. So there was massive dieback of willow trees. It's thought that over 170,000 trees were damaged in this event or died. Luckily, they are resprouting, but the beetles can reinfest those sprouts. So we're still trying to see what's the future in that area. And this is a map of their distribution. Like I said, they have a pretty wide distribution geographically. It's growing pretty quickly, and we had that big dieback here in the Tijuana River Valley, and of course we have tons of different riparian areas that are going to be susceptible to the same thing. So the Polyphagus you can see here is in red, and the Croceo is shown in blue, and then the yellow dots are just where we've deployed traps but haven't detected the beetle. And the beetle was first detected in 2003 here in LA, and then the Polyphagus was, and then Croceo was first detected in the Tijuana River Valley in 2014. And it's thought that naturally these beetles are able to spread about 8 miles per year. So you can see that they've spread quite farther than that since the time that they were detected. And so we're thinking that their dispersal is largely human facilitated, mostly like the spread of infested firewood or infested plant parts. So don't move firewood around. And not much is really known about their natural colonization, why they are colonizing the areas they're in, and how they're moving on kind of a smaller local pattern, how they're dispersing. So this sounds different. So that led me to my question of wanting to understand sort of those local patterns of dispersal and use that to identify areas of high risk and hopefully find ways to prevent beetles from establishing in areas that they are not yet present. So to do that, I have 3 questions. What is the current distribution of shot-hole borer at the Santa Clara River? I'll talk a little bit more about that specific watershed in a bit. What host species are preferred or susceptible, most preferred or susceptible to these pest disease complex? And then what semiochemicals might be effective at deterring shot-hole borers? And I will explain what a semiochemical is later on in my talk. So let's start with this distribution question. So I do a lot of my work in the Santa Clara River, which is a large watershed that spans North LA County to Ventura County. It's highly impacted by urbanization. Here's in Santa Clarita, you can see that there's urbanization right along the edges of the river, which is right here. And then in Ventura, there's a lot of impact of agriculture. You can see the agricultural lands are right next to the river, and there's a lot of runoff and things affecting that area. And now we have the shot-hole borer, which is further impacting the system. So I already had detection traps there, about 42 in Ventura County, but we hadn't really done any detection trapping of shot-hole borer up in LA County, the LA region of Santa Clara River. And so for this fellowship, I deployed 12 additional traps in Santa Clarita to sort of complete our analysis of the entire watershed to make a total of 54 traps. So what I did is I used these inverted 2-liter water bottles, cut out a little window. Here you can see this is a quiciverol lure. That's an attractant that's used for these beetles. The beetles fly in, they fall into this preservative, and I can then sort that sample. And so we deployed the traps in September in Santa Clarita, and we monitored them bi-weekly, just looking for presence and absence. Then once we collected our samples, my great interns would sort through all the other insects that got into the traps. We'd find the shot-hole borers, send them down to UC Riverside to get identified. And right now in Santa Clara River, we've only identified the prolific shot-hole borer. And so here's a map of where we have detected shot-hole borer in the Santa Clara River. So these are all new detections here in Santa Clara. There's actually 3. It's a little difficult to see that one there, but there are 3 new locations that we found them. And as you can see, they're quite widespread throughout the Santa Clara River. However, it's still kind of a patchy distribution. It's not really known why they're not in areas like Fillmore or right over here. So hopefully going to figure out what makes them prefer certain areas over others. So it leads to my next question: what host species are most preferred or susceptible to this pest disease complex? And although this beetle is a generalist, of course it has many host species, it's possible that they have more preferred species than others, right? And it has been shown, actually, some researchers at UC Riverside showed that they do have different preferences and susceptibility between avocado varieties. So what you're seeing here on these graphs, on the x-axis are different avocado varieties. On the y-axis here, you're seeing the number of attacks on those different varieties. And you can see that the beetles did prefer certain varieties over others. And then over here, the researchers measured the number of galleries per surface area, which was sort of their proxy for susceptibility, so how easily the beetles can make a gallery within the tree. And they found that, again, there were differences in the avocado varieties in that susceptibility. And interestingly, the ones that were most susceptible were not always the ones that were most preferred. So that could create some interesting dynamics with riparian species if we could see the same patterns. So I looked at 5 different host species, which are the dominant riparian trees in Ventura and North LA counties. That's California sycamore, Fremont cottonwood, black cottonwood, arroyo willow, or red willow, and arroyo willow. And what I did, this should actually say 10 blocks. I did 10 different blocks with these cages, where in each cage I put 1 branch sample of each tree species. Sadly, I didn't get a nice picture of when I had it all set up. This was after I had opened the box and it had gotten moved around. But when I did set it up, it looked more like this, with the trees or the branches actually standing upright. I placed 50 beetles in the middle. I had a cup of water in each cage to increase relative humidity and promote the fungal growth. And once I released the beetles, I gave them 1 week to choose their species that they liked the best and start creating a gallery, potentially inoculate fungus or lay eggs. Then I destructively sampled all of the branches, which took a very long time, but to measure preference, we just simply counted the number of attacks. So you can see we just counted the holes right here of the beetles, the ones that decided to enter that tree. And then to measure susceptibility, we measured the length of each gallery, so how easy was it for the beetle to make a gallery in that species. We looked at the presence of fungus. You can see here it causes this necrotic tissue, it is staining. So we just looked for presence or absence of fungus. And then we looked for the presence of eggs to see if the beetles were actually reproducing. We did find some eggs. You can barely see them here. They're really, really small. But I actually left that out of the analysis because I thought with the destructive nature of our sampling, we could have lost many of the eggs, and I couldn't really say for sure that they weren't in certain species. Okay, so what did I find? I found that these beetles really are generalists within the riparian species. Here are the 5 species that I tested, and I looked at number of attacks. And although the Fremont cottonwood surprisingly had the most number of attacks on average, it wasn't significant. So they do prefer all of these trees, and they're all going to be attacked by the beetle if it's in that area. I did find potentially differing susceptibility to shot borer, though. So again, you can see the species on the x-axis and then mean gallery length. So it looked like the Fremont cottonwood and the red willow had the largest gallery, so the beetles were potentially more successful in those species. And most interestingly, looking at the fungal growth, presence or absence of fungus, although Fremont cottonwood was the most preferred, it actually was the least likely to have that fungus established within it. So, sorry I didn't explain this graph, but again, it's the species on the x-axis and the proportion of galleries with fungal growth. And you can see that the cottonwoods really had much less probability of getting that fungus actually established within that 1 week period. So sycamore and willows were much more likely to have fungus in those galleries. And so this can definitely affect population dynamics as well as dispersal from certain host trees because the beetles' reproduction is based on the amount and quality of their fungus. So when they have more fungus, they reproduce more quickly, and they also create larger broods of beetles. So it's potentially going to be slower dispersal from these cottonwood species and maybe faster dispersal with sycamores and willows. Okay, lastly, I looked at what semiochemicals are effective at deterring shot hole borers. So first I need to tell you about the control methods that are already in place, and I only have a couple minutes left, so I'm going to go really quickly. The current methods are cultural control, basically physically removing branches or trees that are infested, chipping them up to a size less than 1 inch, and then solarizing that wood. You can probably tell that that's a really expensive process, so not ideal. There's also chemical control. There's many fungicides and insecticides like amamectin benzoate, imidacloprid, and bifenthrin that are they are able to kill the pest disease complex within a host tree as long as it's not already severely infested. But those are highly toxic, especially in aquatic areas like riparian areas, so not ideal. So what about the use of semiochemicals? And don't worry, I'm going to explain what a semiochemical is right now. It's basically any chemical that conveys a signal from one organism to another. So in this case, we're looking for chemicals that convey to the beetle that they want to go away from wherever that is coming from. Okay, and so I looked at 3 different chemicals. Verbenone, which is a known anti-aggregation chemical for bark beetles. Piperitone, which is actually like the inverse chemical structure of quiciverol, which is that attractant. And then ethanol, which is usually an attractant for most ambrosia beetles, but has been shown to potentially deter shot hole borers. And so, I actually just finally started this experiment last week, so I don't have data, but I'll just show you what I did. I used a Y2 bioassay, so it's this small Y plate, which we put 2 different airflows in the top, and you can put different chemicals, and it's basically affecting the beetle's olfactory system. So you put the beetle in, that's a beetle right there, and it's sensing whatever chemical you put in there, and it gets to walk up and make a choice either to the right or left arm. And so that's how they first tested attractants. So they showed that the attractant always took the beetle to whatever side it was in. So the way it would work for me is to see if we can determine if these semiochemicals are effective at deterring shot hole borers. We first put the attractant in one of the arms, and show that the beetle significantly picks that arm over the other arm. Then in the same arm, I would add the deterrent. So I'm basically showing that the deterrent overrides that attractiveness of the quiciverol. And I would either see, if it's an ineffective deterrent, I would either see that it picks the right side now, or its activity is shut down completely and it doesn't even want to walk up because it is deterred from that deterrent. So sadly, I don't have results from that yet, but it is coming soon, so stay tuned. And so today, hopefully I showed you that with this fellowship, I've better characterized shot hole borer distribution at a local scale at the Santa Clara River. I've shown that shot hole borer is truly a generalist within riparian species. It's not really showing preference for any species over another, but host species may differ in their susceptibility to that fungal growth, which could affect population dynamics and dispersal patterns on a local level. And so with this information, I'm going to hopefully identify areas that are most at risk of shot hole borer infestation, maybe areas with more willows and sycamores. And then if I can find effective semiochemicals, deter those in locations that are not yet infested to hopefully keep beetles out of that area. And I just want to end that I actually just learned from Richard Stouthammer, who's also studying these semiochemical deterrents, that some of them were not as effective as they'd hoped in the field. So I'm hoping with my research I can show that maybe those chemicals aren't even effective in the lab, and we should throw them out the door, or maybe we need to adjust our dosages. So I'm going to be testing different dosages of the deterrents. And with that, I will take any questions.
So it's pretty clear in the lab how these semiochemicals are introduced.
Mm-hmm.
How are they introduced in the field? What's
Great
the
question.
mechanism for that?
Usually it's like a little pouch which has a time release. The chemical is somehow time released, and you would deploy it, many of them on a specific tree, maybe a really expensive or important cultural tree, or along perimeter of an area that you don't want infested, maybe green spaces. We can somehow deploy them around that area, but it is like a time release pouch. Yeah. Yes.
What's the cost of the deterrent?
Deterrent? Oh, it's much less expensive than I don't have a number for you, I'm sorry, but I know it's much less expensive than removing trees and a lot less expensive than using those pesticides.
So it'd be feasible to, you apply like on each tree? Or?
Yeah. Yeah. You would want to apply probably multiple on each tree,
Okay.
or in front of the trees even. Yeah.
Okay.
Is
Yeah.
there a minimum branch diameter or a diameter breast height that the beetles won't enter?
Yeah, it's about 2 inches
Okay.
diameter that they usually don't enter. Yeah, which is pretty small. So most trees are susceptible. Yeah.
Yeah, I have a question on your design. So when you put the beetles, it can only attack one bark, or can it go multiple?
So I did account for, I looked for the beetles within the galleries to make sure they stayed there, and that was their choice. And there were a couple times I found 2 beetles in one hole, but I never found a hole without a beetle. So once they made their choice, then that was the tree they stayed in. Yeah.
And then with the semiochemical, is it possible that they just didn't want to go to either? Do they just stay put? Is that like the control condition or something, or I don't know.
But can you?
With the Y tube,
Yeah.
could they just have stayed at the bottom and been like, I don't like either path, or do they typically?
Well, so one's a control and then one's a deterrent.
Oh, that's right.
So,
this is already that right. Okay. Okay.
But potentially they would just have complete shutdown of activity, and that to me would say that the deterrent is effective before they were walking up to the attractant. Yeah.
Is there any precedent for these chemicals working? Because I can think back to LDAM and Santa Cruz and light brown apple moth. I'm using similar strategies to control that. So is there any kind of precedent for these working, or is this completely innovative?
No, deterrents have been used for bark beetles, and that has worked. I don't know much about other species besides bark and ambrosia beetles, but I know it has been successful for some bark beetles.
I was going to say in your Y tube, maybe you should just do just a control, just for a reference point to see.
Oh yeah, I always do a control to make sure there's nothing.
Well, no repellent,
Oh yeah.
So just to get the numbers, in case they don't make the decision to even go, because my experience working with beetles, sometimes they don't do anything.
Yeah, I always do a trial run where there's nothing in either of the arms,
Mm-hmm.
and just see what their activity is, and actually to test whether there's something with my system that's contaminated, or maybe a light source is drawing them a certain way.
Mhm.
So I always do those trial runs
Cool.
first. Yeah.
Okay.
Yeah.
the honeydew, we actually saw the smallest diameter branch down about a 1/2 inch.
1/2 inch. Okay.
Okay.
Well,
Not a lot, but we.
we'll have to update that then. Yeah.
I was curious about the bottle traps that you guys were using.
Mhm.
The lower attachment, the reservoir.
Yeah.
What was that? It was a different type.
It's just a falcon tube. It's really common scientific
Yeah.
tube. But before we were just using it, we would just keep it capped. But we found in the windy areas that it wasn't far enough down that the preservative would spill out. But we really just created that in the lab based on other people's designs. And you don't have to use the exact same thing as long as there's something there that catches them.
It just looked nicer. I've been using the stout hammers for years ago.
Mhm.
The two balls.
Yeah. Oh, with the 3D
Yeah,
printed.
the coupler in the middle and all
Mhm.
that.
Yeah.
The couplers are getting harder for me to find.
Really? Yeah. Well, I mean, you can buy the falcon tubes on the internet.
I do.
Yeah.
It's a little more professional.
Any other questions? Great, yeah,
Thank
wonderful,
you.
thank you. Super, okay.